Okay, welcome to this inaugural Automated Patch Clamp in Academia webinar by Sophion. This is actually a double first, because not only is it our first webinar, but it's our first in a series of academic talks from users and collaborators that have been doing some great work with APC and with other techniques in the world of ion channels. My name's Damian Bell. I'm going to be your host, and we're going to have a great science talk from Dr Nina Ottosson from the University of Linköping, following a brief introduction by myself. I will just pull up my slides, and then we can go into my talk, and we can start the presentation. So yeah, welcome everyone to this webinar by Sophion. My name's Damian Bell, and I'm going to tell you a little bit about Sophion. We're global leaders in automated patch clamps. Some of you may already know, some may not. We make two automated patch clamp machines, the Cube, which has the possibility of 384 simultaneous recordings, and the Q patch, which can do 8 to 48 simultaneous recordings. Despite our small size, we're only a company of 61 employees. We have a global footprint. We've got labs and demo offices across the world, as you can sort of see on this map. And because of the innovative developments and novel technologies that we are creating at Sophion, at least 50% of our time and resources would be put towards R&D in terms of capabilities and assays upon the automated patch clamp machines. Today's introduction is going to be talking about how APC is being used more and more in academia. We've got a number of Sophion APCs in academic laboratories already. So these four examples I will be going through shortly. So the University of Queensland, Yale, University of Sussex, and UC Davis have all got ongoing work with our APCs in their labs. We also have a number of ways that we actually work with academia. So for instance, we've got a very useful program called the Technology Access Program, which would be very useful for any academics wishing to generate their own pilot data for funding, for instance, on a Q patch or a Cube. And that would be a case of actually talking to someone like myself and determining whether your projects would be suitable, and we'd like to almost work with you in that kind of format. There are also obviously ways that we can have collaborations together. So for instance, we've had a very good and ongoing collaboration with the Ashmore and Ahmed labs at UCL and KCL respectively. We also have work ongoing on iPSC neurons with the University of Wisconsin-Madison and a small biotechnology company called BrainCell. And finally, in terms of these examples, we've got the Resin Acid Derivatives work that we were collaborating on with Linköping University. I won't say too much more about that because there's a really nice talk by Dr Nina Ottosson to follow, but just another great example of a collaboration between academia and Sophion. So the first of those examples of academic labs with Sophion APCs actually in their lab and being used to great effect in their research. So the University of Queensland in Australia, there's several strong ion channel labs that have had access to a Q patch based at the University of Queensland since 2011. So for instance, the Vetter, the Lewis, the Craik, the Schroeder and the King labs have all got access to this this Q patch that they have down in Queensland, and all of those labs are studying various different channels and venom toxins, for instance, upon the sodium channels, the calcium channels, and the azic channels. And this is effectively a shared core facility where each of those labs shares costs and experience upon the machine, but also it creates enough and sufficient throughput for them to be using it regularly and routinely in their research, in their academic lab. And it also allows for them to foster really strong collaborations either between themselves at University of Queensland or amongst other labs across the world that potentially want to be collaborating with them in their work and maybe getting access to their QPatch. We've also sort of seen this lab have a really great example or examples of PhD student and postdoc career development having access to the QPatch. So for instance, Julie Clint passed through these labs and is now at Lundbeck using the Cube at Lundbeck. Josh Wingard has again sort of worked in the University of Queensland and had access to the QPatch there. And he's gone on to sort of work for Teva and for now Channel Bio, a small spin out in Sydney, looking at antibodies and ion channels. And again, they've adopted a QPatch in both of those places too. And finally, Christina Schroeder, she's done some great work on the QPatch in Queensland, so much so that she's now moved over to America and she's at the National Cancer Institute, NIH, and she has just acquired a QPatch too for her work at the NCI over in Maryland. These labs at the University of Queensland have really sort of showcased some of the capabilities of QPatch and just how good it is for some of these capabilities. So for instance, because of its throughput, it's a true medium throughput screening capability. So they've screened a number of different toxins and toxin fractions in some of their work. Because it has these microfluidic channels, you can actually apply low volume applications of these very rare and scarce toxin fractions. And also because of the planar array of recording sites, QPatch actually allows very stable and therefore very long duration recordings, which some of these toxins require because they sometimes can be very slow in their activity. So you need a long, stable, high quality recording to make sure that your recording is consistent throughout. And not surprisingly, because of all this work and because of this shared capability, they've published 27 and counting publications using the QPatch alone. And I'll just go through a few of the examples of this. So for instance, they've been using it, as I was suggesting, for some of these ICK or inhibitory cysteine knot-in toxins. So for instance, this top paper in toxins is based on spider-derived gator-modified toxins. And the paper below in JBC is based on conotoxin, so the Conus snail, fish hunting snails. They have a number of toxins that some of you may be aware of, and they've also done a number of studies upon these Conus snail conotoxins using the QPatch. And this is a really interesting publication that's just come out in the last month or two. So this is effectively moving away from the usual spiders, snakes, and scorpions and cone snails, as I was just saying, more into actually trees that have these venoms within their sort of systems. And so this Australian stinging tree gives a very, very painful, very long-lasting stinging effect. And as Nature research highlights in the bottom left corner reported, some of these stinging events can last for months in terms of the potential pain that's developed through them. So there's effectively almost like a neuropathic pain that might be developing in response to stinging from this tree. And some of this data on the right here shows some of the effects of these stinging tree venoms. So another great example that we're very proud to sort of present is just in the last 2 months actually, the Stephen Waxman lab at Yale has acquired a QPatch 2 and a Cube. So this was just announced at the end of September. Steve Waxman's lab, if you don't know, is a world-leading lab in chronic pain. So this is great to have the Cube and the Q-Patch in that field, really driving chronic pain research, as I'm sure the Waxman lab will do. And so the kind of work that they'll be using the Q-Patch and the Cube for are things like chronic pain channelopathy. So the Waxman lab has been doing for years now, decades even, work upon NaV1.7 mutants and defining their biophysics, pharmacology, and also I'm sure doing some drug discovery. And all of this will be possible in a far faster and more efficient form with their Q-Patch and Cube at that lab. Another example of a really fruitful and successful use of Q-Patch in academia is the University of Sussex Drug Discovery Centre. So Professor Martin Gosling and Dr Sarah Lilly were part of the University of Sussex Drug Discovery Centre, and they've now formed a spin-out called Enterprise Therapeutics. And with Sophion, they spent 2 weeks in our labs in Ballarat, and they did a 20,000 Q-Patch compound screen in that 2-week spell. And this was looking for TMEM16A, which is an ion channel, and they were looking for TMEM16A potentiators for the treatment of cystic fibrosis. And this was a very successful screening process because these TMEM16A potentiators entered clinic, and they were getting some very nice results in their early phases of clinical trials. And as a consequence, Rush Genentech have now acquired this portfolio of TMEM16A potentiators for the potential treatment of cystic fibrosis. And another academic Q-Patch that we've actually got out in the wild is at the UC Davis. So some great work that was done in George Chandy's group with Christine Beaton and Heike Wolf at UC Irvine, looking at autoimmunity and the ion channels involved, namely KV1.3 and KCA 3.1. And this was actually something that Professor Heike Wolf has now sort of taken on into her own lab at UC Davis. And just to give you a quick plug for a Biophysical Society networking event that we at Sophion are co-hosting with the Biophysical Society next week, Tuesday, 8th of December. Please sign up if you've got an interest in careers in ion channels, careers in automated patch clamp. We're going to have some great speakers, and we're going to hopefully have plenty of time to have questions and general networking around this, about just how you can potentially sort of use your ion channel experience and be taking it into a career further down the line. So by all means, sign up for that. That's next week, Tuesday, the 8th of December. And this is now sort of a different way of potentially sort of having access to automated patch clamp via Sophion. So here we've got a really nice collaboration with Jonathan Ashmore at KCL and Amir Ahmed at UCL. And so this is something that we call a TAP project, or a technology access project, or technology access program project, sorry, which allows Sophion to have collaborations and essentially access to our machines. And this gives academics the ability to get data, generate data on our machines, and effectively perform proof of concept studies in their own research, and potentially take this on so that they could then use it as data for funding proposals later on. So this proof of concept collaboration has been ongoing for the last 4 years or so. As a consequence, we've got a number of publications on ion channels in cancer, and including in those publications, both Herbert Olsen and Naya Sørensen from Sophion, because we were key collaborators on these papers with them. And we've also got a very nice poster presentation that the Ashmore and Ahmed labs have produced as well. This is all to do with Wnt signaling and ion channels in cancer. And the ultimate aim of this is to potentially provide data for a funding application, with an ultimate goal of having a shared core facility that UCL, KCL, and other ion channel labs, potentially around London, may well be able to sort of tap into as part of this shared core facility that they're hoping to fund through a lot of the data generated through this TAP project access. Not surprisingly, because of this uptake in academic use of Sophions APCs, there's a very long list now, or growing list of academic publications using the Q-patch or the Cube. So if you go to our website and you plug in APC in academia, you can pull up all these publications, and you can also find a lot more information about how you might be able to sort of, for instance, get a collaboration with Sophion and get access to some of these APC capabilities for your own research in your own labs. So a quick summary before I pass over to Nina's great work. APC in academia, we really sort of feel that academia can make a great deal out of the capabilities of APC, because it really will push your research further, faster and more efficiently. Just like manual patch clamp, you'll get high quality gigaohm seals, but you'll also couple that with very high throughput that's capable on our machines. The microfluidics channels that we've got built into our recording plates are low volume, and they have fast exchange of test solutions. You can also potentially do temperature control on our machines. Current clamp is also another possible mode of recording upon the machines. And all of this data, all of these capabilities fully complements potentially your manual patch clamp data generation within your own ion channel labs. And we do realize that obviously one of your biggest hurdles in terms of actually getting an APC in your lab is the fact that you may not have access or have data on these machines in your particular ion channels or your particular fields, which is why we have things like the Technology Access Programme to allow you to potentially sort of generate that data for funding proposals. Or another way of actually getting that kind of access is to collaborate with us at Sophion. And this collaboration could be with us at Sophion, but also it could potentially be with other academics and institutions in terms of a shared core facility consortium that you may be able to get access to. And of course, if any of you do have any ideas or proposals, questions around these kinds of uses in your own research, please do contact me. There's my email. Please feel free to send me questions, ideas about how you might be able to sort of work with Sophion, because we're really keen on getting our APC machines into more and more labs to really further ion channel research as fast and efficiently as we can do. And again, you know, another great resource potentially would be to go to our website, put APC Academia into the search box, and that should bring it up. That's enough of me talking. I'm going to now pass over to Nina at the University of Linköping. She's got some great work. I will just pull her slides up, and I'll give you a quick introduction to Nina. Nina did her PhD in the Fredrik Elinder lab at Linköping University. She was looking at shaker KV channel openers. That work has now been extended into KV 7.2, 7.3 channel openers. And she also had a really nice collaboration with Fredrik Elinder and with Sophion, looking for these kinds of channel openers on the Q-patch. And she'll now tell you far more in detail about some of their great work that they've been doing in this area. Over to you, Nina.
Thank you. And first of all, I would like to thank Sophion for organizing these webinars, and also then giving us the opportunity to present some recent data from our project. So I work as a researcher in Fredrik Elinder's lab, where we then work with these resin acid derivatives as openers of the KV 7.2, 7.3 channel. And we're also now investigating whether these compounds have the possibility to be developed to antiepileptic drugs or not. So let's see if I can change. Good. So today, there's more than 50 million people worldwide that suffers from epilepsy. And more than 30%, so more than 1/3 of the patients still don't get seizure-free despite an optimal treatment. And during an epileptic seizure, there are groups of neurons that are firing too easily. And this can, for example, be caused by damage of the brain or either then by malfunctioning ion channels. And the ion channels, they are obvious targets for antiepileptic drugs. And as a rule of thumb, the excitability can either then be dampened by blocking of voltage-gated sodium channels or by opening of voltage-gated potassium channels. And many of the classic antiepileptic drugs, they do act then by blocking voltage-gated sodium channels. And we are then working with an alternative strategy, then that would be to open voltage-gated potassium channels, and more precisely then the Kv7.2, 7.3 channel. And also I just have to mention that I don't have any pointer, so I will try to guide you through the slides and where to have a look on them. But I think that should work. So our target is voltage-gated potassium channels that has 6 transmembrane domains. So segment 1 to 4 builds up the voltage sensor, and segment 4, 5, and 6 the pore domain. And it takes 4 of those to then construct the functional ion channel. And in S4, we have a number of positively charged residues, mainly arginines. And these arginines, they then detect and respond to changes in the membrane potential. So when the cell is closed and the inside is negatively charged compared to the outside, S4 is positioned in a down state close to the intracellular side. But when the inside instead gets positively charged, these moves upwards then. And this movement then causes a conformational change that will open then the ion conducting pore. And our target, the Kv7.2, 7.3 channel, or it's also called the M channel, it is encoded by the KCNQ genes. And this family belongs to the family of Kv7 channels that has 5 members, so from Kv7.1 to 7.5. And some of them can exist as homomers, but the channel that is our target then is the Kv7.2, 7.3. It is as a heterotetramer. So it consists of subunits of both then Kv7.2 and 7.3. And this ion channel is expressed throughout the neuron, and it has a very specific voltage dependence. So it's actually open to some extent already when the cell is in rest. And this gives it a very unique and important properties to really then set the resting membrane potential and thereby also then set the excitability of the neuron. And this Kv7.2, 7.3, it is actually the target of the antiepileptic drug retigabine. So retigabine was approved in 2011 and it was made by JSK. And retigabine shifts the voltage dependence of activation for this channel furthermore to negative voltages. And this will cause that the resting membrane potential will be hyperpolarized, and also then in some types of neurons that will also have an effect on the after hyperpolarization, as we can see here on the action potential. And then this will then dampen the excitability. And as a result, then this will then prevent this high frequency repetitive firing that is connected then to an epileptic seizure. And the effect of retigabine is very well studied, and it's known to bind to a tryptophan that is found on S5. And this tryptophan is conserved among Kv7.2 to 7.5. So in addition to the target that is then the 7.2, 7.3 channel, it also opens then, for example, the Kv7.4. And in contrast to the antiepileptic targets, Kv7.4 is expressed in smooth muscle cells. So opening of this ion channel will cause vasorelaxation. And this is considered to be the reason to one of the adverse effects of retigabine, that was urinary retention. So then opening of Kv7.4 channels in the smooth muscles of the urinary bladder then caused this urinary retention. So retigabine had several drawbacks and had also then a limited use, why GSK stopped the production of it in 2017. But it had a very good antiepileptic effect. So despite all this adverse effect, it really validated the Kv7.2, 7.3 as an antiepileptic target. So there are, luckily enough, several pharmaceutical companies and also many then academic groups that are searching for other compounds that could then be developed into new type of antiepileptic drugs. And more precisely, we are searching for compounds that target the voltage sensor of this ion channel. And we would like these compounds to have a reduced effect then on these smooth muscle cells compared to retigabine, and also then that we would like them to have antiepileptic effects to start with in a very simple in vivo model. And these are two of the aims that I will describe for you here in the presentation today. So in Fredrik's lab, we have for quite a long time been working with the movement of the voltage sensor. And we know that different compounds that have a lipophilic part, so for example, then different polyunsaturated fatty acids and also resin acids. And so compounds that are lipophilic, as we can see in their black parts, and also then that have carboxyl group, for example, like these ones have here, as we see in red, so that these compounds can be negatively charged at certain pH then. So we know that these compounds, they bind to the ion channel in a position that is very close to the voltage sensor, and thereby it electrostatically attracts the movement of the voltage sensor. So it really facilitates the upward movement of the voltage sensor. And as Damian said, I studied the effect of these resin acids on the shaker channel during my PhD studies. So then we, for example, could find out that on the how it really affects the movement in detail then. So here to the left, we see the voltage sensor domain of the shaker channel. So in blue sticks, we see the position of the different gating charges. So the uppermost is then R1. And the figure next to it, we see the position of R1 during activation. So in addition to moving upwards, S4 actually also rotates upwards. So then we see how R1 translocates from C4 to C1 in the closed state, and then also then to the open state. And it's actually this last rotation of movement that then is facilitated by our resin acids. So in addition to this, we could also find that they do bind to a lipophilic pocket that is formed between S3 and S4 helices, and then the lipid bilayer. So from this position, the negative charge of these compounds will have a direct interaction with the uppermost gating charge to have this effect then. So it is actually compounds that act and opens the Kv7.2, 7.3 channel by these mechanisms that we are searching for. And in the lab, we don't have any APC. So our primary assay is instead the 2-electrode voltage clamp technique. And it is maybe not as fancy, but it has a lot of advantages. So we use oocytes from the Xenopus laevis frog, and we inject small amounts of RNA. So during a couple of days, the oocyte will express millions of copies of the ion channel. And then, thanks to the size of the oocyte, so they are more than 1 millimeter in diameter, we can insert 2 electrodes into the oocyte, so we can then clamp the voltage and then record both the voltage and the current. So for our recordings, we use voltage step protocols. So we step the voltage from minus 120 up to then approximately plus 30 millivolts. And after these steps, we then make it a step to minus 30 to record the tail currents. So first we do the recordings in the control solution, and then manually by a syringe, we apply the compound to the oocyte containing well and do then the recordings in the presence of the compound. And well, many of our compounds, what they do is that they then shift the voltage dependence of activation, as we can see here down in the GV curve. And actually, this ion channel has a very slow kinetics, and also it does not inactivate. So therefore, we use the tail current as a good measurement for the conductance. And we have been studying the effect of the resin acids also then on the Kv7.2, 7.3 channel. And so we have published that DHAA, that is the naturally occurring resin acid, it does not act as an opener at physiological pH, but when we replace the carboxyl group with permanently negatively charged sulfonic acid, as we can see as U161, and also then introduce this sulfonic acid at a stalk of 3 atoms away from the 3-ring structure, we actually generate quite a good opener of our target. And maybe I should also mention here that the naming of these compounds, that is thanks to the name of our chemist. So his name is Wu. And so he has synthesized a couple of derivatives here now. So Wu161, when we clamp the oocyte to minus 50 millivolt, it actually increased the conductance more than 7-fold. And if we look on the GV curve instead, we can see that it shifts the voltage dependence of more than 20 millivolts to more negative voltages. And also then, if we look on the more positive voltages, we can see that in addition to that, it also increased the maximum conductance of this channel. But for this mechanism, it's actually most the voltages, the more negative voltages that are important. So if we look on the dashed line for the GV curve, we can see that it's the opening almost only then comes from this shift in voltage dependence. And we have earlier also shown that the compounds, they need to be negatively charged to act as openers. So when we remove the negative charge, we also remove their capability of acting as openers. So these data are already published then for the Kv7.2, 7.3 channel, but all the rest of the data that I will show you today will actually be unpublished. So in addition to analyzing then the effect of the heteromere, Kv7.2, 7.3, we have also tested the effect of 161 on the homomeres, so Kv7.2 and 7.3. And it's not possible to do recordings of Kv7.3 as a homomere. So then by just introducing a point mutation that is found on the pore domain, then you can do recordings of 7.3 as a homomere. And we can actually see that 161 induce a shift that is very similar on all these 3 channels. So also then at concentrations up to 30 micromolar, there are no differences between the heteromere and then the homomeres. And for us, it's of course very important to investigate that these effects that we see, that they are not dependent on the expression system. And in particular, since we believe that the membrane is a very big part of the binding pocket, so then it's of course extra important for us to investigate. And therefore, we have done collaborations, or in the collaboration with Sophion, we have tested our compounds then on the targets, so the Kv7.2, 7.3 channel expressed in the cell line, so using the APC. And then we can see that there are no differences in the affinity between these systems here. Just checking there, but okay. So, so we have investigated the binding sites for these compounds to improve them as openers, starting with the binding site of retigabine. So if we look at the first GV curve, we can see that retigabine induces a shift that is quite big on this wild type channel. But when we remove the charge or the tryptophan here, we can see that the effect of retigabine disappears. And then when we make this test also our compounds on this channel, we can see that there is no difference whether we have this tryptophan or not. So we can see that I see that I have some questions here. Oh, it was quite a long one. It's been shown that retigabine at concentrations below. So I just had a question here. I guess you all can see it in the chat function. Thanks for asking here. So it says that it has been shown that retigabine at concentrations below 10 micromolar causes a strong decrease in the rate of deactivation with a minor effect on voltage dependence. Do you know how resin acids affect the activation? Well, I only have preliminary data for that. And well, they do have some effect on the deactivation kinetics, but it not seems to be as pronounced as it is on the as by retigabine, I would say. But it's really ongoing project, so maybe we can answer that in more detail in slightly later. And also then, how does resin acids affect resting potential? So quite long time ago, we made recordings on GRG neurons, and then we could see that also our resin acid derivatives had some effect actually on the resting potential. So then let's continue here with the binding sites. So in addition then to the retigabine binding site, we have also investigated one of the binding sites that been shown to be very important for the effect of different benzamides. So for example, then the compound called EK73. So these residues, as you see in the green blob here, they are found on the more intracellular side of S3. And EK73 is actually a very effective opener of the Kv7.2 as a homomere. And when we mutate this residue, the effect disappears completely. So in the middle GV curve down here, we cannot even see the control anymore because it's just below the green curve. But just for the retigabine site, there are no differences between the effects for Wu161 when we make these mutations. So this then strongly indicates that Wu161 does not act then via the retigabine site, and it does not act via the benzamide site. But instead, we had from our recordings on the shaker channel, we knew that the resin acids have a direct interaction with the uppermost gating charge. And the Kv7.2, 7.3 is actually very similar to the shaker channel, and in particular at this part. So therefore, we investigated how a neutralization of the uppermost gating charge affected the effect of the 161. So as we see here on the middle GV curve, we can see that 161 can still open and shift the voltage dependence of this mutated channel, but the shift is reduced to less than half. So this strongly indicates then that we have a direct interaction with R1, but even when we neutralize it, we still can see some effect. And that could, for example, be that we have an interaction with R2 instead when we remove R1. So this, in combination with what I said earlier, that we need the compound to be negative charged to act as opener. So then this really strongly indicates that our compounds, they do act by the desired mechanism that we would like them to have on the voltage sensor. So we have made quite a few derivatives. So in addition to investigating the charge of the stalk, we have also just tried to see if the atom here in the stalk have any makes any difference. So, but when we just replace the oxygen with the nitrogen, we can see that this does not affect the binding at all. And from earlier, we also know that different halogenations can have a very beneficial effects for the compounds as openers. So we have worked with that as well. And for example, generated the compounds called Wu199 and Wu200, where 199 has a bromine and 200 has a chlorine at another position. And these halogenations, it really improves the compound as openers. And actually, there are no differences in the effects between Wu200 and retigabine. So we can see also here from the GV curves that they look very similar in all aspects. So as we said earlier, that for us it's very important then to be able to predict the potential vasorelaxing effects of our compound. So therefore, we first investigated the effect on the Kv7.4 expressed in the oocytes. And retigabine is known to act as an opener of this. So here we have the GV curve for the effect of retigabine. But in contrast to all the data that I've shown you earlier for for the GV curves, we can now see that the most of the opening effect does not come from a shift in voltage dependence, but instead it comes from an increase of the maximum conductance. So in the next curve, in the concentration response curve, we see the effect of the shift and voltage dependence for the different concentrations. So we can see that for all concentrations, the shift induced on the Kv 7.4 channel is smaller than the shift induced on the Kv 7.2, 7.3. But then in contrast, when we instead look at the ratio of Gmax, we can see that at the Kv 7.2, 7.3 channel, there are no effects on the maximum conductance, but instead then on the Kv 7.4 channel, retigabine induced a very large increase of the maximum conductance, supported in at 30 micromolar, but also at more physiologically relevant concentrations, starting already at 3 micromolar. And for Wu200, it looks instead extremely similar to the Kv 7.2, 7.3 channel. So this compound, it causes a leftward shift of the voltage dependence. And we can see if we look on the middle graph on the second row there, that there are no differences at all between the two ion channels. And also then when we analyze the range of Gmax, there are also no differences between the ion channels. So for us, this was very interesting, but also then we need to be able to really know how these different effects could affect then the vasorelaxing effects. So then we started a new collaboration with Tom Jepp's lab in Copenhagen, and together with them, we could test then the effect on tissue. So for these recordings, they worked with freshly isolated mesenteric arteries from rat. And what they do is that then they measure the constriction or the relaxation then of these tissues. So here we see two recordings, and on the x-axis we have the time, and on the y-axis we have the constriction force. So first, what they do is that they pre-constrict the tissue, and then by applying increasing concentrations of retigabine, they can see that this will then cause a vasorelaxation. And then they do the same recording again, but in the presence of linaprazine, that is a Kv7 channel blocker. And now we can see that higher concentrations are needed to cause the vasorelaxation. And this can also be seen then in the difference of the two curves in the concentration response curve to the right. So this shows that yes, retigabine, as already known, has these vasorelaxing effects that to some extent at least is caused by an effect on the Kv7 channels. And when we make the same recordings with Wu200, we could see that also this compound has caused some vasorelaxation, but we can see that the effect is smaller than for the retigabine. So it takes higher concentrations to cause vasorelaxation than for retigabine. And this is, of course, very promising for our compounds. So this will also then be continued to investigate in more detail. And as Damian mentioned, we have a collaboration with Sophion where we have tested the effect on different ion channels. So for us, it's been very important to investigate other types of voltage-gated ion channels, since the voltage sensor is very conserved among the different voltage-gated ion channels. So for example, we have investigated the effect of the voltage-gated sodium channel 1.5. And in contrast to our target, this channel will inactivate very quickly after opening. So therefore, for us, it was very important to investigate the potential effect, both then on the activation, but also on the inactivation. And we could see that starting with Wu161, we had no effect on the voltage dependence of activation. And for the inactivation, we could actually see that we had a leftward shift. But that was only then at high concentrations. So for the other compounds investigated, as we can see now on the second row here, we did not see any effect on the activation, and it was only then at this high concentration that we saw this shift of the inactivation. So in addition, we also investigated on the APC the effect on the voltage-gated calcium channel 1.2. And just like the sodium channel, this inactivates. But here we could not see any effect on the activation. So on the inactivation, we could see a small shift of 210 micromolar, but this effect was not dose dependent or concentration dependent. And lastly, we also investigated the effect of compounds on hERG channel. And it's the same here, that at concentrations up to 10 micromolar, we did not have any effect, and neither then on the shift of activation, and also then neither on the maximum current of this on the ion channel. So altogether, these data then really supports that these compounds have a fairly promising voltage selectivity profile. And also then, as I mentioned, that we would like to investigate if our compounds have the possibility to act as have an antiepileptic effect. And we did this using zebrafish larvae. So zebrafish larvae that were 3 to 5 days post-fertilization. And these larvae at this age, they are known to express our targets, so the Kv7.2, 7.3 channel. Though we have to keep in mind that these, at this point, these larvae, they do not have a fully developed blood-brain barrier. But for us, that was maybe a good thing actually for us at this point. So what we do is that we insert an electrode into the brain of them, and thereby we can record then the field potentials. So first we see a recording in a control solution, and we do see some activity, but when we apply PTZ, that is a very classic chemoconvulsant that will induce epileptic seizures, we can see that the activity is increased a lot. And here in the orange box, then where you can see what we have classified as an epileptic event. And in the bar graph on the second row, we can see that the frequency of these epileptic events is then increased multifold by application of the PTZ. So then we validated our method, so partly then with carbamazepine that did not have any effect, and also then that with valproic acid that could prevent epileptic seizures, and most importantly then that also retigabine could prevent the seizures in our zebrafish larvae. And then we also, of course, then investigated our compounds using this method. And before we did these recordings, we also just tested that the concentrations that we applied were not toxic for them, so that there were not more reduced activity due to toxic effects. So we investigated Vo161 at concentration of 10 micromolar, and 199 and 200 of 10 and 30 micromolar. And we could actually see that at all these concentrations, all the compounds actually had an antiepileptic effect. That, of course, is very also promising for our project. So that was the last slide with the results that I have here today. So just a quick summary, what we have said today then. So that is that our resin acid derivatives, they do act as opener of the Kv7.2, 7.3 channel, and they do that by an electrostatic mechanism having a direct effect on the uppermost gating charge. And we could also see that the halogenation was very beneficial for the effect. And even though our compounds, they do have an effect on the Kv7.4 channel, so they shift the voltage dependence of it. But despite that, it seems like they have a smaller vasorelaxing effect than retigabine has. And then from the APC recordings, we could see that they had almost no effects on these different cardiac ion channels. And we could also then see that they could prevent epileptic seizures in zebrafish larvae. So we consider these resin acid derivatives as being a very promising scaffold for a new type of antiepileptic drugs. But of course, we're not there yet. So for the moment, we are working very much on to find a more detailed binding site. And that is, of course, then partly then to improve their affinity for the target, but also then to be able to avoid an effect on the other Kv7 channels. And today we are supported by a drug discovery and development platform in Sweden. So they help us also then to improve different pharmacological properties of our compounds. And we would also, quite soon, also would like to test an antiepileptic effect of our compounds in a slightly more complex model than our zebrafish. And I would also like to take the opportunity to now to say that we are really looking for collaborators for future development of these compounds. But in addition to these resin acid derivatives, we're also working with other chemical scaffolds in the lab that are unpublished. So please contact us if you have some questions or suggestions there. So with that said, I would just like to thank the people that have been involved. So first of all, my colleagues in Fredrik's group, with the people in blue are the ones that have contributed a lot to this presentation. So, and Urban and Michelle did the recordings on the zebrafish, and Argel and Malin for the oocyte recordings. And in particular also to Wu, the chemist, and to Tom and Salome in Copenhagen for doing the recordings. And Ulrika for a lot of discussions about these chemical properties. And also a special thank to Melanie at Sophion for the recordings. Thank you.
Great talk, Nina. Thank you very much. Beautiful GV curves all over the place. And as you'd expect from the Linda lab, very, very thorough. Thank you very much. That's really good. A few housekeeping points. If you do have questions for Nina, I know there's one from Alex, for instance, that I'm going to give to Nina shortly. Please put them into the chat box. Also, before we go into the questions, there is, as part of the chat box, a poll area. If you could just take a few minutes to answer some of those questions in the poll, that'd be great. And now on to the questions, Nina. So you've answered one of mine, for instance, which was, how did you come up with the naming system? So it's obviously your great organic chemist, Wu. Yeah. So congratulations to Wu too, because that's some really nice chemistry there that you've obviously sort of done a lot of work upon, the ion channel sort of drug discovery areas that you've been working in. One question from Alex was, are these acid resins voltage dependent in their activity?
Well, yes, I would say so. Well, voltage dependent in, they will change the voltage dependence a lot. And if he means that if they bind, if whether the channel is closed or not, that is something that we are working with for the moment. But I would say that we think that they are very close to the binding site also when the channel is closed. But for us, it's since it is an electrostatic interaction, the interaction will be the larger than to the higher kind of the position of the voltage sensor. So in that case, that would be that they have voltage dependent effects.
Okay, Alex just had a couple more follow-ups, which sounds like you've just about answered it as he was typing it, because you were talking about the voltage sensor and its position in the membrane. And I think that's almost what Alex was driving at with his question. So
where do I, where are his, here they are. Oh, as they're changing. Oh, sorry, I didn't see them. As they changed. Yeah.
Yeah, so I think you almost answered Alex's question very thoroughly in the end. But Carlos has come back with another question. Can you see that,
so
Nina?
What is the effect of the drug when you hold the membrane potential at more physiological resting? So, so if we hold the cell at the voltage that will keep it closed, then like -70, and if we apply the compound, then we will see an increase of the current.
And while we're waiting for some more questions, okay, so we've got another one from Carlos.
Great. So
Carlos is peppering you with questions at the moment, which is great.
Really good. So the washout, I would say that for these compounds, it's fairly okay. So for the compounds that are permanently negatively charged, they are fairly easily to be washed out. But then for some of the other compounds, as I showed, like for DHAA, that are not permanently negatively charged, then we see that the washout is not as good. So it differs a bit between the chemical properties for the different compounds. But very good question. Thank you.
And there's another one just popped up from Ahmed.
Have you tried these compounds on other KV channels? Yes, we have of course worked with them a lot on the shaker channel, maybe to extremely detailed, let's say, on the shaker channel. But in addition to that channel, so we have not worked with them so much. We have tested a few of them on KV 2.1, but I have to be honest, I think it 8 years ago, so I don't remember the results, to be honest.
And another one from Carlos. PIP2.
I wouldn't think so, since the effect of PIP2, it's really needed for it to kind of stay open, PIP2. So I don't think that it works. No, I don't think so. That it can compensate for PIP2, no.
And while we wait for another question, I was going to ask you about, because these are lipoelectric effects, yes. Presumably the lipid composition of your membrane may well have an effect on the lipoelectric effect of these resin acids. Have you seen any differences between, for instance, oocyte membranes and mammalian membranes, for instance, because obviously they'll have different lipid compositions. I mean, in terms of oocytes, you even have very complex folded architecture of the oocyte membrane. So do you have any data that shows difference between mammalian versus oocyte?
That's a very good,
activity?
That's a very good question. And because since we think that the lipid membrane is almost a part of the binding pocket, so it's a very extremely important question for us. But as I show data for Vo161, both from APC and the oocyte, and for that compound, the affinity are identical. So for some of the compounds, it differs a bit, but they are almost maybe surprisingly similar, I would say, for these two. From these two, at least, yeah.
Okay. Yeah, I mean, as you almost suggest, because of their mechanism of action, you'd almost think that they would be very sensitive to different lipid compositions that oocyte versus mammalian might have, but apparently it's not sufficient to be making a big difference in terms of these compounds' activities. Another one that you almost touched upon is because they're charged compounds, will you do you think you're going to have problems, for instance, with the blood-brain barrier?
Yes. Yeah.
Okay.
So actually, these compounds that I show you data for today, they are permanently negatively charged, and we have done so. We don't think that these compounds will be able to cross the blood-brain barrier. Then that is partly then from in silico studies, but also then in vitro studies that suggest this, that they will not be able to cross the blood-brain barrier. So that is kind of one of the chemical properties that we are working with to improve then to have effect, but still be able to cross the blood-brain barrier.
So something that your brilliant chemist Wu needs to work upon then is to make them blood-brain barrier penetrant.
Yep. That's a little challenge for him, but we will. I think we kind of have our suggestions how to manage.
You've got some ideas of how to solve that. Okay, great. There's another one from Carlos coming.
Yeah. Sorry, I believe I missed this. Do these drugs affect? So what happens when you add KCNQ? So I was almost waiting for that question. So they do also open KV 7.1. I have not tried KV 7.1 without KCNE1, but it's possible that it could change the effect. Maybe it depends on which compounds we talk about. But also, I mean, since it's not on the retigabine site, then they also act on that channel. Your pick. Thank you.
Okay, Nina, thanks for your talk. That was great. Really, really interesting stuff. And again, some beautiful work that you've been doing there at Linköping University. I think we should potentially call it to an end. If anyone does have any final questions, last chance to ask the expert right here, right now. Otherwise, by all means, send me questions and I'll relay them to Nina if need be. But yes, more questions, more ideas, more thoughts. Send all your questions to me and I will contact Nina accordingly. But thanks again, Nina. That was a fantastic talk. And that brings the first inaugural Sophion webinar to a close. Thank you all for your attention. Thanks for your time. And yeah, please do fill out the poll questions if you get a chance before you log off. That'd be lovely. Thanks again, Nina. That was perfect. Thank you. Bye then.